this. And because I know we all have a time.
Yeah.
So, and I respect
No,
time.
no, you're good. Thank you.
Yeah.
Oh, you need coins? Grab your coins. Head on out.
You ever written down? But that was a good favorite then, and I'm like, that will definitely take a.
Yeah, it's perfect. Okay, what have you got?
We got these for these.
Yeah, he was the one leaving messages, right?
All right, and that's our completed.
All right, I will check this in a few minutes, and then I will probably be able to hand you phase 2, which will make me super happy.
How many phases are they?
9. But some of them are gonna move way faster than others.
I'm gonna use your microwave real quick.
Yes, of course you may.
I didn't know if we did it. It was the grandma. She had a tire arm. Dawn to 11. Yeah. Yeah. No, there was obviously the grandma. They weren't here last year, but it was a nice. On say. What's that? Wait, no. Oh, this is the crime scene. Or is that the time? I know. Yeah. Hey, that's looking. We kept the same crime scene.
I did not have, that might have been my sister. It's blonde and I was around. Yeah. But that was actually. Hanging out there.
All right, are we ready? Okay, let's go ahead and get started. So we have a little time at the end of class. I'm going to roll through this quickly because I know you guys want to get back to your cold case as much as you can. So I think you only had the one because you were quiet today, friend. All right, here we go. So we're going to talk about how we identify the substance of genes. What are these things and how do we figure it out? So we started this process by working with bacterial transformation. So in the middle of the 1900s, scientists were asking questions about genes. So what's a gene made of? How do genes work? How do genes determine the characteristics of organisms? So here's what they knew from the beginning. They knew that a section of DNA on a chromosome, a gene or something like a gene, whatever it is on a section of DNA in a chromosome in a nucleus. And they knew that chromosomes contained hundreds of these genes. And they knew that genes contained most of your characteristics, like your hair color, your skin color, how things taste to you. Who here likes cilantro? Who here thinks it tastes like soap? If you think it tastes like soap, you have the original historical gene. If you like it, you have a mutated gene. Happy to be a mutant because I really like me some guacamoles.
If you can't taste it, deteriorate.
That's probably genetic too. That's interesting.
Soap is a cat and it tastes slightly like soap, but for the most part, I can't taste it.
Can't taste it at all. That's interesting. Yep. So a lot of this is genetically determined.
No, you have to eat soap first so you know what it tastes.
There you go. So the genes contain the instructions for making specific proteins because all these things like how you look, how your body functions, they're all based on protein. And that the genes determine the order of amino acids in making proteins. So we started with Frederick Griffith in 1928. So he started this process with bacterial transformation. So he injected mice with how many samples of bacteria? Do we know, Colin? 4, but good, close guess. 4 samples of bacteria. He had heat-killed disease-causing bacteria, which means you heat it just enough so that you keep its structure or whatever, but it's no longer going to infect you. And he injected mice. Yes, what you got for me, Lucas? I mean, Xander? Yes, they lived. Very good. All right, so these heat injected, Yusuf, The heat injected bacteria, when injected into mice, the mice lived. So this live harmless bacteria was injected into mice. What happened, Eunice? And they lived, the live harmless one, they lived. And then what caused the fatal pneumonia? Do we know? So the fatal pneumonia was caused when you put the two strains together. So you take a strain that used to be able to cause disease, you heat kill it so it can no longer cause disease. Then you take a strain that does not cause disease. You put the two of them together and somehow you're starting to get disease being caused. So something's happening, something's being exchanged between these two organisms. So he concluded that genetic information could be transferred from one bacterial strain to another bacterial strain. And so transformation is when one type of bacteria, which is harmless, gets changed permanently into another that is a disease-causing form. And the transforming factor must be a gene. So in summary, it looked like this. We're going to get an ad. Let's see what it is today. Oh,
pizza.
Klarna. Always Klarna. Now we're talking about pizza. How appropriate.
That's when you probably.
So we got pizza in our lab, right?
Is the default, and pay later is there for when you need
Do, 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 do.
That's
Klarna,
That's
Klarna
right.
killed her.
Not Klarna.
Griffith is thought to be the first person to demonstrate and document the process of bacterial transformation. Griffith conducted his famous experiments while attempting to find a vaccine for pneumonia. Today, we can treat most cases of pneumonia with antibiotics, but in 1928, pneumonia was often deadly, even in otherwise healthy people. The Streptococcus pneumoniae bacteria comes in several strains, and Griffith experimented with two of them. He used a virulent S strain with a smooth polysaccharide coat and a non-virulent R strain with a rough coat. The coating on the smooth strain protects it from the body's immune system and thus allows the bacteria to attack cells and cause disease. While observing the pneumococcus bacteria, Griffith noticed that sometimes the non-virulent R strain would revert into the virulent S strain. He theorized that a transforming principle was somehow transferred between the S strain and the R strain, causing the R strain to acquire dangerous properties. To test his theory, he injected 5 types of bacteria into groups of mice: a live R strain, a live S strain, heat-killed S strain, a combination of heat-killed S strain and live R strain, and finally S strain that had been isolated from mice in the 4th group. The results of Griffith's famous experiment would confirm the presence of a so-called transforming principle. Mice from group 1 who were injected with the non-virulent R strain survived, which is unsurprising given that the R strain is not dangerous. Also unsurprisingly, the mice in group 2 who were injected with the virulent S strain developed pneumonia and died. High temperatures kill most germs, which is why boiling something is often a cheap and effective way to sterilize it. Griffiths used heat to kill S strain bacteria, then injected these dead bacteria into a 3rd group of mice. Even though they had received a dose of the dangerous S strain pneumococcus bacteria, these mice survived because the bacteria were all dead. The 4th group of mice was crucial to Griffith's experiment. He injected the mice with a combination of heat-killed virulent S strain and live innocuous R strain. The mice injected with the combination developed pneumonia and died. On their own, neither of these components should have been deadly, so Griffiths concluded that the live R strain had acquired some type of deadly component from the dead S strain. He called this mysterious component the transformation principle. Finally, Griffith isolated bacteria from the blood of the 4th group of mice and found it to be identical to known samples of the deadly S strain pneumococcus bacteria. He confirmed this by injecting the isolated S strain into a 5th group of mice, which then developed pneumonia and died. The live R strain had transformed into S strain by acquiring some tiny dangerous properties from it. At the time of his experiment, the exact nature of this transforming principle was unknown to Griffith. It took almost 20 more years before Avery, McLeod, and McCarty's work in 1944 confirmed that nucleic acids and not proteins are the molecules of heredity. However, Griffith's groundbreaking work proved that some organisms can acquire new properties from their environment and from one another, and also that non-heritable exchange of genetic information is possible. If you have questions about Griffith's experiment or about bacterial transformation, please check out the comment section, as viewers often ask and answer one another's questions in there. Feel free also to check out the study guide that's linked in the video description. Thanks again for watching, and please remember to like, comment, and subscribe.
All right. So, kind of a simple experiment, but it gave us a lot of information. This is our guy, Frederick Griffith. Yay.
Yeah.
All right, and then we have Oswald Avery's experiment. Truthfully, this involved more than Oswald Avery, though he's the one that gets most of the credit these days. So his transformation, he discovered that DNA stores and transmits what you get, Barrett? Yeah, genetic information from one generation to the next, and that DNA is responsible for transformation. And Oswald Avery, Colin MacLeod, and Macklin McCarty, where do you suppose those guys were from?
McDonald's.
Scotland, right? These are some Scottish dudes. So basically what they did is they went through the process of elimination. They knew that there were certain things that were part of these single-celled organisms, these bacteria. They had proteins, lipids, polysaccharides, RNA, DNA. So they put things in that would basically chew those up and mess them up and screw them around and make them not do what they're supposed to do. And by process of elimination, they determined that it was the DNA that was passing along the information. So I'm not going to show you this whole video for the sake of time, but it's in your references. But it basically shows the step-by-step of how they did this. And here's where they ended up. They had a control where they added nothing. They removed the proteins. They removed the RNA. They removed the DNA. Not until they removed the DNA did they find out, oh, we've done this process of elimination. No longer passing along genetic information when there's no DNA there. Nope, move. And these are those guys. So this is Colin MacLeod, Macklin McCarty, and Oswald Avery. Those are those fellows. So then we have these things called bacteriophages, and these are bacterial viruses. Believe it or not, that's actually how they look. I know it looks mechanical. It doesn't look like something that would occur in nature. It looks like a spaceship or a lunar lander or a robot. They're cool, y'all. They're super freaking cool.
Built with a light bulb.
So they are viruses that infect bacteria, bacteriophages. So all bacteriophages are composed of what kind of molecule that's surrounded by a certain structure? Do we know? Does anybody know this one? So they have a nucleic acid, so genetic information, right? Covered by a protein. Basically, up in that little compartment at the top is where they store all their genetic information. And it's like a little protein envelope that holds it in. And then they quite literally land on the cell and inject it. It's so cool, right? So a bacteriophage attaches itself to a susceptible what? What do they infect? What do they infect, Xander? Yep, bacteria. Bacterium, if we're talking about one little cell, and they infect that host cell. And then following the infection, the bacteriophage hijacks the bacterium's cellular machinery to prevent it from producing bacterial components and instead force it to produce what? What's a virus force a cell to produce? Lucas. I couldn't tell whose hand that was. Both of your hands are up. Was it yours, Lucas? It was yours. Okay. More viruses, right? Viral components. So eventually new bacteriophages assemble and burst out of the bacterium. And what do we call that when something breaks open? Anybody remember the word for
Vision.
Not fission. That could be, do you know?
Separation.
So lysis is what we're looking for. Lysis. It lyses. The cell lyses. It explodes and it spits out a whole bunch more bacteriophages. Bacteriophages occasionally remove a portion of their host cell's bacterial DNA during the infection process and then transfer this DNA into the genome of a new host cell. So what do we call that process? When the bacteriophage is going to take some of the cell that it infected, take some of its DNA, and then take it with it
Pollination.
it sounds like pollination, right? But this is what we're calling transduction. All right, so that's what's happening there. So they're going to take a little bit of that cell's DNA or genetic information in whatever form it's in, and take it with them to the next cell they infect. So bacteriophages only inject DNA, not proteins, into bacterial cells. And this we know because of Hershey-Chase. So now we're in 1952. So Hershey-Chase experiment studied a bacteriophage composed of DNA and a core protein coat. They wanted to determine what part entered the bacterial cell. Is it the protein coat, this part up here, or the DNA inside that protein coat? So they used radioactive markers to label the DNA and the protein. So that is super cool. And this is kind of what that looked like. I love how they show a little blender here. They blended it up. But in the end, they were able to determine that you're only getting nucleic acids, and those are your molecules of heredity, not these proteins. I will leave this one for you to watch on your own as well. I want you to understand that they worked with the bacteriophages, but for the sake of time, we'll move along for those. All right. So this is Martha Chase, Alfred Hershey. So these are the Hershey Chase people. Yes.
They're like couples.
No, just
They aren't.
colleagues.
Why are they all named after food items?
Yeah, interesting. So what's the role? Yes.
Are viruses a single cell organism? Like, are they actually?
No, viruses are not alive. A virus is not alive. A virus carries genetic information, but it's not alive. It cannot be killed. You can denature the protein envelope in a virus, which will make it non-functional, but you can't kill it because it's not alive. So everybody that talks about, like during COVID, we'll kill the virus by wiping down. Technically, that isn't correct language. You can't kill it because it's not alive to begin with. And when we start talking about viruses and infections and diseases and things like that later on, we'll talk about viruses are very, very primitive because they're like kind of the first things that contain genetic information. But even more primitive than them are these things called prions, and they're real fun to learn about because they are crazy. There's still a few of them out there that can infect you, and they are bad, bad, bad news, like wild bad news. But interesting, very interesting. So DNA makes up genes and must be capable of storing, copying, and transmitting all genetic information in a cell. So number one, under bacterial transformation, what happened when Griffith injected mice with pneumonia-causing strain of bacteria that had been heat-killed? Kenna,
Nice.
the mice survived. What happened when Griffith injected mice with a mixture of heat-killed pneumonia-causing bacteria and live bacteria, the harmless type, Claire?
You died.
They died. Dead mice got pneumonia and died. What was the purpose of Oswald Avery's experiments, Jordan? Yeah, he wanted to find out which molecule in the heat-killed bacteria was the most important for transformation. What experiments did Avery do, Lucas?
Different enzymes that broke down different parts of the cell.
Yes, he used enzymes that destroyed various molecules in the heat-killed bacteria, including lipids, protein, carbs, and RNA. And transformation still occurred until he destroyed the DNA, right? So what did Avery conclude, Eunice?
Concluded that DNA is the molecule that transfers.
Yes, the DNA is what's transmitting your genetic information. And what did Hershey Chase conclude? Nobody's got this one? What did Hershey Chase conclude? Oh, Claire's reading it. Hang on, she's looking. I love it when your own handwriting is good. Yeah.
I wrote it.
Yeah, okay. All right.
Something only transports DNA.
Yeah, the DNA must be genetic material because the phage injects the DNA, not the protein. So how did Hershey Chase confirm Avery's results, Eunice?
Hershey and Chase's experiment proved that DNA only carries it.
Right. So Avery said that DNA transmits genetic material from one generation to the next. And Hershey Chase concluded that the genetic material in the bacteriophage was DNA, not protein, which confirmed Avery's assertion. So for these over here, your labels should be for, this is Hershey Chase. So they had DNA with a radioactive label. The bacteriophage infects the bacterium. Radioactivity now enters the bacterium. Then over here, you have a protein. See, now here they've put the protein with the radioactive label. They inject that into the bacterium and no radioactivity inside the bacterium. Yes.
Is there like a short for the radioactivity?
A way to write the word?
Yeah.
You can come up with whatever abbreviation. There isn't really one, but if you find one that works for you, I have all kinds of abbreviations that work for me. My shorthand, nobody else could understand, but I understand it. So whatever works for you. So yeah, so you can see the little dots, the dots designate that they tagged the DNA with the radioactive label versus tagging all the rest of it, the protein with the radioactive label. And then they were able to determine what entered the bacterium. So I'll put this one up instead of going through it because I can never remember if I'm going left to right or up and down with my animation. So I'll ask the wrong questions. But DNA performs three functions. It's got to store, it's got to copy, and it's got to transmit. And you could obviously have this store and transmit flip-flopped right or left on yours, it doesn't matter. Just be sure that you have that correct. So understoring, it's important because DNA makes up genes that control development characteristics. And an example might be instruction to cause a cell to become a giant oak tree would be written in your DNA. And then for your transmitting information, it has to be able to pass it from one generation to the next. And an example might be that your gametes of the oak tree carried genetic information from parent to offspring so that the offspring also develops into an oak tree. Why is copying important? Because genetic info must be accurately copied with every cell division. And your example might be that after mitosis, each new oak tree cell has the same genetic info as the parent cell. I'll give you a second, make sure everybody has that. All right, well, what's the structure of DNA? All right, so here's what they knew going into the process. This was a hot topic. There were a lot of people working on this. A lot of people wanted to find this out because this was like the thing. This was the hot topic in science. They knew going in that DNA had certain components, including a 5-carbon sugar, which is deoxyribose, a phosphate group, 4 nitrogen bases, which are your nucleotides, which are purine, which is adenine and guanine, and your pyrimidines, which are thymine and cytosine. They knew all this. So going into it, you have Edwin Shargraf in 1949. Yeah. Oh, yeah. So in DNA, there's always equality in quantity between bases A and what?
G and C and A and T.
Yeah, G and C and A and T. Okay, why is this important? Say your study, you know that all the components that we just listed. You know you've got 2 pyrimidines, 2 purines, you know you've got deoxyribose, you know you've got nucleic acid bases, you know your structure, you got a phosphate group, and you're finding as you analyze the molecules that you always have the same amount of A, of adenine, that you have of thymine, and the same amount of cytosine that you have of guanine. What could you infer? If you know those amounts of A and T are always equal and the amounts of G and C are always equal to one another. Different molecules, they'll be different, but to one another, they're the same.
So they can be copied.
Can be copied, maybe. Maybe. What do you think?
They come in pairs.
They come in pairs. Big. That's a huge, huge revelation. If the quantity of A and T in a particular molecule are always equal to one another, and the quantity of G and C in a particular molecule are always equal to one another, then you know they're in pairs. That's huge. So now we know they're in pairs. And that's this guy, Shargraf. Yay, Shargraf. He discovered that. They came in pairs. And we're like, good. Now we're getting somewhere. So now we get to one of the most notorious scandals in the history of science. Who did physical science with me? Who remembers Edison and Tesla? Remember we went so far as to electrocute an elephant to try and prove who was right? Yes, it was insane.
Well, I didn't do lab rats.
Yeah, you did junior, but yeah, we didn't really talk about it too much in juniors because electrocuting elephants is kind of hard for juniors. But you guys know, we talked about it, and we talked about who did it, who electrocuted the elephant.
Me.
Edison electrocuted an elephant, right? Because he was trying to prove. Do you remember that? What?
That Tesla's electricity was more harmful.
Yes, because Tesla was all about the direct current. He was trying to prove that yes, very good.
Didn't Edison also burn down Tesla's?
Potentially. Allegedly, he potentially was involved in the destruction of Tesla's laboratory. Lost to history, but a lot of people think he did it. Okay, so here is probably even bigger scandal, many people would think. And this is a scandal that remained a scandal and remained on the down low until about 25 years ago. So it was just kind of recently put right. So this right here, this adorable little cherubic face, belongs to Rosalind Franklin, who was born in 1920, July 25th, 1920, in London.
No offense, but he looks like a girl.
She is a girl. Yeah.
The math is.
There's she. That's Rosalind Franklin.
She's
She's
looks like a she.
He kind of looks like a she. Hey,
she.
she's cute. Anyway, so as a young child, she showed extraordinary academic talent. She was very, very smart. She went to this place called Norland Place School, And eventually she got promoted to St. Paul's Girls' School. And this is important because girls were not taught physics and chemistry. Wasn't a thing. You aren't going to need that, dear. Pat you on your pretty little head and go learn how to embroider. You don't need to learn physics or chemistry. Just be pretty and smile, right? Okay, right. So this is one of the few places where she could learn it, and she desperately wanted to learn it, so she goes to St. Paul's Girls' School. Hooray for St. Paul's. Then she went on to Newnham College, and in 1941 she graduated, and they gave her what's called second-class honors. What's the degree you get after 4 years of college called? A bachelor's degree. Those were not for women. Sorry, you can't get one. Sorry, girls. No bachelor's for you. You get second-class honors. Isn't that nice? Doesn't that have a great ring to it? Second-class honors. Such a ring to it, right? So, despite it all, she said, fine, forget it. I am going to apply to Cambridge University and I'm going to get my PhD, and gosh darn it, she did. And her doctoral thesis was called The Physical Chemistry of Solid Organic Colloids with Special Reference to Coal. She spent years looking at coal under fancy microscopes, okay? And this is 1945. I want you to keep this date in your head. 1945, she gets her doctorate degree, all right? So she's young at this point. She asked a friend, keep an eye out for a job opening for someone who is a physical chemist who knows very little of physical chemistry, but quite a lot about holes in coal. She was kind of being self-deprecating. She knew a lot about physical chemistry, but compared to some people, I guess she thought she maybe wasn't up to snuff, but because she'd spent so much time studying the holes in coal molecules. So that's what she did. And she got her first position at King's College. Very good place to work. She worked in what's called X-ray crystallography. So what she did is an X-ray was shined at this little LED screen with one little tiny hole. And one little ray would pass through the little tiny hole and it would hit a sample. And the sample would contain a molecule she wanted to make an image of. And then it would kind of diffract within that sample like a prism. And it would take a picture or make an X-ray photo of that molecule. Something as small as a molecule she could make into a larger photo. Kind of. So she worked in the field of X-ray crystallography and X-ray diffraction, seeking to produce photographic evidence of the physical structure of various molecules. So we had some idea how molecules were shaped based on how atoms interact with one another, based how bonds are. If you remember, those of you who did chemistry with me, you might remember that, there are certain ways to draw models of molecules, like you have this molecule, you know what I mean? So basically what she did is she was taking pictures of them because she wanted to prove it. And then there were some molecules we didn't know how they were structured, one of which was DNA. So in 1951, she had a student working under her whose name was Raymond Gosling. So she's the PhD. He is a PhD, a graduate student. He's working towards his degree. So now he has to work in someone's laboratory. So he captured what has been called one of the most important photos ever taken in the history of man, ever taken. And it's called Photo 51. And that year at a research presentation, she described her findings. And she said, the results suggest a helical structure which must be very closely packed, containing 2, 3, or 4 coaxial nucleic acid chains per helical unit and having the phosphate groups near the outside. What you're seeing, if this is a DNA, a strand of DNA, helix, helix, helix this way, it's looking down it that way. You're looking this way, like a cross section. So what you see there that looks like an X, those are actually the nucleic acids in the middle of the helix. And that's just the one that she got the on top. And then the blur around the outside, you can see that's the phosphate and sugar groups that run down the backbone. And that's what I'm wearing. I'm covered in Photo 51 today. So yay for Photo 51. Now, meanwhile, while she's working on this, at the same time, there are multiple people trying to achieve this goal, right? A lot of people want to figure out the structure of DNA. So you have these two fellows here, and they're at King's College, which is like, these are neighbors, okay? They're working, they're colleagues, they know each other, right? And Franklin left King's College for Birkbeck College, and around the same time, James Watson and Francis Crick were working at Cambridge. So she had her photo, she finished her research. She's like, okay, this project's done. I'm going to go to another school. I got another position there. So she moved over there. These guys continued working. All right. And they were working at the same time that she had been. So they've been working all the way up to the point where she left. Now, they had come very close to creating a model. So while she was trying to discover the structure of DNA by taking an actual photo, like an image of it, an X-ray image of it, they were trying to determine the structure of DNA by using a molecular model. Like I said, we know the way certain atoms angle when they attach to one another. So they knew kind of like these puzzle pieces have to fit together this way, kind of like Legos. So they were kind of putting stuff together to try and figure out what worked. And Franklin had been staunchly anti-model. She said, no, you should only create a model after you know what it actually looks like, as opposed to trying to guess what it looks like by creating a model that works, right? So here's the thing about Franklin. Rosalind Franklin was not warm and friendly and lovey-dovey and warm fuzzy. She was very cold. She was very abrasive. She stared people in the eyes when she talked to them, and it unnerved people. It made people really uncomfortable. She was just not, probably not somebody you'd be like, oh my gosh, I want to be her friend. You know what I mean? Like, just not a warm, welcoming person. That's difficult enough. Somebody like that, to be around somebody like that or work with somebody like that is difficult enough. But when that person is female, it adds another level of complication, because remember, she wasn't even supposed to get the bachelor's degree. You know, you don't need to learn science. You're pretty little, don't worry your pretty little head over science, that kind of a thing. So smile, ladies, smile, smile. She wasn't for that. She wasn't doing that. That was not her jam. Like, she just was not. So because of that, that made it even worse for her. Okay, it complicated things for her. Now, James Watson later wrote that he and his male colleagues referred to her as Rosie. And that is the name that the people at King College London used behind her back. So that's not her name. Her name's Rosalind. And she didn't like Rosie because she had a great aunt that had been called Rosie. She didn't like that. Her family members, if they ever shortened Rosalind, they just called her Roz. She didn't like being called Rosie, and they did it anyway, even though they knew she didn't like it. So they were poking the bear a little bit here. She made it clear to an American visiting friend whose name was Dorothea Rocky, while sitting at Crick's table at the Eagle Pub. So this is the place where they'd all come out, and Crick was always at this table, and she was sitting there at Crick's table. So Crick was likely there, and all of these people that work in this field, even though they're competing with one another, they're still hanging out together. And this person said to her, Dorothea said, well, how do you like to be called? And she said, I'm afraid it will have to be Rosalind, and added, most definitely not Rosie. That was a pointed remark to all of those men who she knew was calling her Rosie behind her back. Like, I don't like it, knock it off. Do you suppose they quit? No, they didn't. So during this very contentious time period of competing research, Raymond Gosling showed photo 51 to this guy named Maurice Wilkins. And Maurice Wilkins took that straight to Watson and Crick. He is your spy in this story. So Gosling showed it to Wilkins. Wilkins took it to Watson and Crick. Now Watson and Crick have the final piece of their puzzle. They go, oh, looking at this, it's twisted. Okay. And then they put their model together. Look what we did. Yay, we did it, right? But upon seeing the model, Franklin just said, it's very pretty. How are they going to prove it? Right? That was it. Watson and Crick submitted their research for publishing just after Franklin submitted hers. Here's the thing, in science, in medicine, if you want your research to become like officially recognized, or basically to have any clout whatsoever for your research, it has to be published. There are multiple publications, small publications, big publications. When I was in college, we all had to have one published research before we left. I have published research. It's in one little small thing, but it's published. It's a thing. So theirs was being published in a big scientific journal. But she submitted hers, then they submitted theirs. But when they were putting together the research journal, the publisher put Watson and Crick's research first and Franklin's after theirs. So that made it look like she had confirmed their work rather than they had used her work to complete their work. So the credit for decades went to Watson and Crick. No credit to Franklin. She just got swept under the rug like she didn't even do it. And then she died. 1958 at the age of 37. What do you suppose? What do you suppose?
She died.
What disease do you suppose?
Uh,
No, Drew?
STD?
No. Yeah,
Pneumonia.
not pneumonia.
Cancer.
Cancer. How did she spend her days doing what?
Brady.
Radiating things, right? She had all kinds of X-ray around her all the time. She,
Did you say STD?
yeah, he did. It was a good guess. I mean,
She's-
back in the day.
If she's like not very warm, then
That could be a thing. Yeah.
The way you made it sound, you were like, what do you suppose? Think it's probably something dirty.
No, it wasn't anything dirty. No, it was cancer. It was cancer, ovarian cancer, which almost to me is like, that's almost like another jab, because it's like she had to fight so hard because of her gender and where she was. And then it's a reproductive cancer that got her in the end. So to me, it's sort of like some cruel, poetic, I don't know. I don't like it. I don't like it at all. But anyway, that's what got her. Now, one other thing that complicated her life. Look closely at her tombstone. What do you notice? There's rocks.
Julian.
Say it.
Julian.
She's Jewish. How do you know? There's Hebrew there, and the rocks, which are also a thing that are often done with Jewish graves, just placing little rocks with prayers. She's Jewish. So she was female, Jewish, unfriendly. None of these things worked in her favor.
You got
Right?
nerfed 3 times.
Yeah,
She got nerfed.
yeah, none of these things worked in her favor. So when you put all of that together, you end up with somebody who was fighting an uphill battle and didn't make things any easier for herself by not being friendly and not smiling and whatever else. So she, not like she should have had to. But anyway, she died at 37 years old, which is very sad because she could have probably accomplished a lot more. So the Nobel Prize in Physiology or Medicine in 1962 was jointly awarded to Francis Harry Compton Crick, James Dewey Watson, and Maurice Hugh Frederick Wilkins for their discoveries concerning the molecular structure of nucleic acids and its significance for information transfer in living material. All right, well, now we know that they got their big clue from her. So shouldn't she get a Nobel Prize? Does she? Why can she not get a Nobel Prize? They're never given posthumously. So she's dead. No Nobel Prize for you, Rosalind. Sorry. Done. Just like that. That's her legacy. Done.
Their names are so big.
Yeah, their names are so big. Now, if you care to look it up, there's a great TED Talk that James Watson did. And it's from about 15 years ago, I think. But he's active, he's on stage, and he's talking about, oh, and Kirk and I, we were doing this, and he wrote a big book about all this too. But he was carrying on about discovering the structure of DNA, and he was so cool. And at one point, he brings up Franklin, and he's like, people try and say that she did this and did that, but really, he, even now, this many years later, just swept her contribution right under the at a TED Talk. I watched the TED Talk once. I was like, what did he just say? And I rewound it. I couldn't believe it. Like, even now that we know we have the data to support the historical line of things, he still won't admit it. So I'm not sure that I'm a big fan of him, but yeah. Anyway, so the positioning of the molecules. So what photo number? Caitlin. Photo 51. And what was the guy's name? Her lab assistant was Raymond Gosling. You can remember Ryan Gosling. There you go. But not that cute, unfortunately. He was kind of a derpy looking dude. He really was. And he's a spy. So the diffraction pattern determined the helical nature of what? What was the Escher?
Yeah.
There you go. Yeah, of the double helix is basically what it is. It determined that it was a double helix and that it was anti-parallel. Outside of the DNA chain was a backbone of alternating deoxyribose and what? You know?
Question.
Oh, what's your question?
So was photo 51 named after it was taken in 1951?
No, it's just that that's the number they got to. They were taking multiple photographs.
That's my mom's photo.
No, nothing to do with Area 51, but that's how I remember it too, because I don't remember numbers well. So the backbone of DNA is composed of deoxyribose and phosphate groups, okay? The base pairs, the order of which provides codes for the what building? What's built? What do we build? We build proteins, right? And thereby inheritance are inside the helix. There's Rosalind, poor Rosalind, 37 years old. Maurice Wilkins, our spy from now on. And then Watson and Crick using their evidence from Franklin's photograph to determine the structure of DNA. And there they are. Well, look at us and our cool model. I actually love that model.
Dude,
Yeah, no.
what dungeon are they in?
These guys, yeah, look, they're posing everything.
They look like they're in
Look.
dungeon.
Take our picture. We're getting a Nobel Prize. Right? We got Nobel Prize for this. All right, so this is a good summary of that entire crazy ordeal.
When buying snacks for the road.
Good
you
Lord.
pay right away.
Why are these such long
But
commercials
paying
too?
for a new windshield, that's when you probably pay later. With the Klarna card,
Yeah.
debit is the default, and pay
Debit.
later is there for when you need it, so you never lose
Wait,
control.
why is it debit?
That's smart, that's Klarna.
Credit card.
No, they're different.
The discovery of the structure of DNA was one of the most important scientific achievements in the last century, in human history, in fact. The now famous double helix is almost synonymous with Watson and Crick, two of the scientists who won the Nobel Prize for figuring it out. But there's another name you may know too, Rosalind Franklin. You may have heard that her data supported Watson and Crick's brilliant idea, or that she was a plain-dressing, belligerent scientist, which is how Watson actually described her in The Double Helix.
I'm telling you, he's a great guy.
Thanks to Franklin's biographers, who investigated her life and interviewed many people close to her, we now know that that account is far from true, and her scientific contributions have been vastly underplayed. Let's hear the real story. Rosalind Elsie Franklin was born in London in 1920. She wanted to be a scientist ever since she was a teenager, which wasn't a common or easy career path for girls at that time. But she excelled at science anyway. She won a scholarship to Cambridge to study chemistry, where she earned her PhD, and she later conducted research on the structure of coal that led to better gas masks for the British during World War II. In 1951, she joined King's College to use X-ray techniques to study the structure of DNA, then one of the hottest topics in science. Franklin upgraded the X-ray lab and got to work, shining high-energy X-rays on tiny wet crystals of DNA. But the academic culture at the time wasn't very friendly to women, and Franklin was isolated from her colleagues. She clashed with Maurice Wilkins, a lab mate who assumed Franklin had been hired as his assistant. But Franklin kept working, and in 1952, she obtained Photo 51, the most famous X-ray image of DNA. Just getting the image took 100 hours. The calculations necessary to analyze it would take a year. Meanwhile, the American biologist James Watson and the British physicist Francis Crick were also working on finding DNA structure. Without Franklin's knowledge, Wilkins took Photo 51 and showed it to Watson and Crick. Instead of calculating the exact position of every atom, they did a quick analysis of Franklin's data and used that to build a few potential structures. Eventually, they arrived at the right one. DNA is made of two helicoidal strands, one opposite the other, with bases at the center like rungs of a ladder. Watson and Crick published their model in April 1953. Meanwhile, Franklin had finished her calculations, come to the same conclusion, and submitted her own manuscript. The journal published the manuscripts together, but put Franklin's last, making it look like her experiments just confirmed Watson and Crick's breakthrough, instead of inspiring it. But Franklin had already stopped working on DNA and died of cancer in 1958, never knowing that Watson and Crick had seen her photographs. Watson, Crick, and Wilkins won the Nobel Prize in 1962 for their work on DNA. It's often said that Franklin would have been recognized by a Nobel Prize if only they could be awarded posthumously. And in fact, it's possible she could have won twice. Her work on the structure of viruses led to a Nobel for a colleague in 1982. It's time to tell the story of a brave woman who fought sexism in science and whose work revolutionized medicine, biology, and agriculture.
It's time to honor Rosalind L.C. Franklin, the unsung mother of the double helix.
She's just so tragic. It just makes me so sad, you know, to do all that work to fight that entire battle only to get screwed over by the system and die of cancer. I mean, geez, it's awful. All right, so here's what your double helix model looks like. So this is kind of what Watson and Crick were working on. It is double-stranded, a double helix with side ladders that are composed of your sugar group, which is deoxyribose, and a phosphate group, held together by hydrogen bonds. You have steps within the ladder. Those are composed of your purines, adenine and guanine, and your pyrimidines, thymine, and cytosine. So now we know how all that's organized. So the building blocks of DNA are, yes, nucleotides. You got it. Yep. Nucleotides in DNA are made of 3 basic components called a sugar called what? What's the sugar called? Jordan. Deoxyribose and a nitrogenous what? Eunice. Bases. Yep. All right. A phosphate and nitrogenous bases. Yep. Okay. DNA contains 4 kinds of nitrogenous bases. Everybody say them together. What are they? Yes, adenine, thymine. Look at those. They didn't copy over. They'll show up at some point. In DNA, what is joined in any order? What can be joined? What you got, Eunice?
Nitrogen.
The nucleotides. They can go in any order. That's what gives you your different. That's what gives you your different combinations of genetic information, right? Nucleotides and DNA are joined by what kind of bonds, Caitlin?
Covalent.
Yeah, covalent or, yep, very good. Okay, now I'll show you these. Chargraph, he knew that your percent of A and T are equal and your percent of G and C are equal. Franklin, her diffraction photo revealed the double helix. Watson and Crick built your first accurate model. Well, they did build the first accurate model, so we can give them credit for building the first accurate model. Of course, they got a little bit of help from somebody in her photo. So if your percent of adenine is 31.5, then your percent of thymine is going to be, you said 31.5. And that tells you that guanine and cytosine are going to be what percent, Eunice?
Guanine and cytosine are going to be.
Yeah, 18.5 and 18.5, right? Okay, so how about this one? If you've got 20% and 20% in your guanine and cytosine, 30% of your thymine, how much of your molecule is going to be adenine? Barrett? 30, very good. All right, now if you've got 17% cytosine, how about your adenine, thymine, and guanine? Anybody else? Jordan?
I know guanine 17.
Yep. So 17 and 17. Here's how you figure this out. We're going for percent. So we need a total of, say it again, 100.
28.
17 plus 17 is?
34.
Okay, 34. So how much is left over? 100 minus 34?
64. 67.
67 divided by, well,
66.
66, sorry.
33.
Divided by, there you go. Okay, so there we are. So 33, 33, 17. Does everybody understand how that worked? Nope. You need 100% if you know 1, and there are 4, and they go in pairs. If you know 1. So let's make it really simple. Let's just say you have 10% adenine. Then what percent thymine must you have? 10, because they're always equal. What's our total for percent? 100. So if we add our adenine and our thymine, 10% and 10% give us how much percent?
20.
20. So 100 minus 20 is 80. So we know that the guanine and the cytosine have got to equal 80, and they're going to be an equal amount. So they're each what? 40. Good job. All right. And I'll show you this one so you can see it. Hydrogen, there you go. You can have a look at that. Make sure that you get those correct in your diagram. All right. So how does it replicate? How do we copy the code? DNA has to get copied before a cell can divide. It occurs during late interphase or S phase. It unzips into 2 strands. Then you have 2 complementary strands that get produced, and each new copy has 1 original strand and 1 new strand. Then you have DNA polymerase, an enzyme that joins individual nucleotides to produce a new strand of DNA. It looks like this, and this is probably the best way to explain it is this little video. It shows it perfectly. It does. It kind of starts loud, so just when she talks, she's like, "This is a cell." And then
DNA is a molecule made up of 2 strands, twisted around each other in a double helix shape. Each strand is made up of a sequence of 4 chemical bases, represented by the letters A, C, G, and T. The 2 strands are complementary. This means that wherever there's a T in one strand, there will be an A in the opposite strand. And wherever there's a C, there will be a G in the other strand. Each strand has a 5' end and a 3' end. The 2 strands run in opposite directions. This determines how each strand of DNA is replicated. The first step in DNA replication is to separate the 2 strands. This unzipping is done by an enzyme called helicase and results in the formation of a replication fork. The separated strands each provide a template for creating a new strand of DNA. An enzyme called primase starts the process. This enzyme makes a small piece of RNA called a primer. This marks the starting point for the construction of the new strand of DNA. An enzyme called DNA polymerase binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to the 3' end. One of the new strands of DNA, the leading strand, is made continuously. The DNA polymerase adding bases 1 by 1 in the 5' to 3' direction. The other strand, the lagging strand, cannot be made in this continuous way because it runs in the opposite direction. The DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragments. Each fragment is started with an RNA primer. DNA polymerase then adds a short row of DNA bases in the 5' to 3' direction. The next primer is then added further down the lagging strand. Another Okazaki fragment is then made, and the process is repeated again. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase enzyme then fills in the gaps that are left behind with DNA. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. DNA replication is described as semi-conservative because each DNA molecule is made up of one old, conserved strand of DNA and one new one.
So your lagging strand is made in chunks, right? And we call the chunks what?
Oh, August.
Okazaki fragments. Your leading strand is made kind of continuously, one by one, but your lagging strand is done in chunks. All right, so replication in living cells. In prokaryotic cells, cells without a membrane bound nucleus, right? There's only one point of origin. Replication occurs in 2 opposing directions at the same time, takes place in the cytoplasm. Now, in eukaryotic cells, which is our focus, we have multiple points of origin and use unidirectional replication within the nucleus of the cell. Remember her?
No.
This one's great. Miss Sloan's got a good one here.
Guys, this is called DNA replication. So this is gonna be our DNA. Start with supercoiling, and we're gonna use topoisomerase to relax the supercoiling. Then we're gonna unwind and unzip using helicase. Then we're gonna keep the two strands apart using what? SSB. Then we need to lay down primer because we have a leading strand and we have a lagging strand. And the leading strand is considered a leading strand because it's the 3' end of the sugar, right? Whereas the lagging strand is the 5'. So we're gonna use RNA primase and lay down primer one time on the leading strand, and then primer, primer, primer on the lagging strand repetitively. Then we're gonna use DNA polymerase 3, who's gonna work off of the primer and go boom, 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 right down our leading strand, then boom, 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 and those are called Okazaki fragments. Then we need to get rid of that primer that's laid down, so we're gonna use DNA polymerase 1. We only have one primer to remove here on our leading strands. We go primer, then we go primer, primer, primer. Now we need to close the phosphate sugar backbone to seal it. So we're going to use ligase. Then these double-stranded DNA, and this is called semi-conservative replication because we have an old strand and a new strand. These are gonna twist up and form sister chromatids held together by a centromere, and they're gonna separate during anaphase.
There you go. She did it once again. She's great, man. All right, so why are strands of a DNA molecule said to be complementary? Barrett. No? Lucas?
Each strand can be used to make the other strand.
Each strand can be used to make the other strand. Perfect. What is the first step in eukaryotic DNA replication? Kenna.
The DNA molecule separate into two strands.
Yeah, it's got to unzip. Yep. If the base sequence on a separated DNA strand is CGTAGG, what will be the base on its complementary strand? Claire. There you go. And what enzyme joins individual nucleotides to produce a new strand of DNA? Eunice.
DNA polymerase.
Yes. Okay. Now, I messed up with the homework and put 2 different options. Basically, you could hand in the crossword puzzle or you could do the online download upload. Either will be acceptable. So if you saw both and were confused, either is acceptable. If you did the hand in crossword puzzle, it looks like this. All right, so before we go on and work on dragons or labs or whatever you choose to do with your remaining time, we got Miss Macrina for a few minutes. I'm going to swap over to her screen. Do we have anybody absent? I don't think we do we? Let me make sure that I'm sharing the right screen, though. Can I share it so that it records? Maybe it won't. We just share screen.
I did both.
Yeah? All right. You're fine. Okay, and then your... Okay. So if y'all would please give your attention, you can step on this side and then just tap to click. If y'all would give your attention to Miss Macrina, she's giving us an extra special lesson on all of her stuff that she does well. This is about media literacy because she has to do a project and with a teacher, so I suggested media literacy.
Hello, my name's Macrina. I'm a graduate student for the online University of Alabama Library and Information Sciences. I thought I'd present to you guys about how you can find your resources and advocate for media literacy. So we're going to be discussing the type of resources you guys would need for your unit for studying and research purposes. What is media literacy? The 5 components of media literacy. Ignore that last part.
That's fine.
Okay, so since you're studying genetics right now, I thought 2 resources that would be helpful in case you have no idea, like a certain answer or a question. And if Danny isn't available, then you can also look at these 2 sites called the Biology Corner or Google Scholar. They are very helpful. If you have a Google account or a Gmail, then you can get inside more detail on Google Scholar. Now, what is media literacy? It's the ability to access, analyze, evaluate, create, and act using all forms of communication. And as a librarian, it's our job to spread awareness and educate you guys on how to do your research properly and not use researcher bias. When it comes to media literacy, it's all about using our critical thinking skills. Like, when did this specific post or article come out? Who made the post or who wrote it? And why did they write it? And we're addressing the bias because it's everywhere, including social media. And some social media sources can be false. And you have to
No.
be, yeah, and you have to be aware of AI as a source. It can be all fun and games, but sometimes it's used as a lying purpose. Now, what is what is the difference between misinformation and disinformation? Misinformation is a false or misleading information. It means that they shared it without any harm. They weren't thinking. It was all an accident. But disinformation is spread to mislead. It's deliberately shared to spread hate, create all it's all on purpose. Examples of misinformation are like clickbaits, fake studies and statistics, or satire mistaken as fact, like the Bumble. It's all a joke, but people take it like so seriously. It's all a joke.
Yeah.
Yes, and then manipulated media or deep fakes. And examples of disinformation are biased news sites, conspiracy theories, bots and trolls that are spreading false narratives. They're doing that on purpose to make you angry. But how can we combat this? It's all about fact checking and make sure the sources are credible. And media literacy education should start in like late middle school, early high school. And we have to verify before sharing stuff on social media and use fact checking algorithms. And Google Fact Check Explorer is an excellent example. It's a tool for teachers and students can use to find debunked news reports. That's how we break the bias. These are my references.
Are those credible?
Are those credible?
Yes, they are. Yes, they are. They definitely are. That's a good question.
So she gave me a whole bunch of links and I've already made a file in your resources folder with all of the links that she's provided, which are great because you can use them. For sure, it's stuff like when you're doing your final project. Great idea to look.
And the media literacy articles would be very helpful so you can spread it to everyone else and
Yes.
make sure it can help. It will help you apply for your personal life and your education.
Mm-hmm.
It will definitely help.
And it's getting harder, right? I mean, how many videos have you seen that you had to watch 10 or 20 times before you could convince yourself it was AI? Especially the week, the newer, I mean, sorry, yeah.
That's how they get you with news.
Yeah, that's how they get views. Mm-hmm.
Yep.
There's some that you look at and you're just like, I think this is fake, but I don't know why. Have you seen one of those yet?
I love showing my grandma GTA and like making her think cars can fly.
I saw one where it was a bunch of people on a bridge collapsing. It looked like China, like in the mountains.
Oh my God.
And this bridge collapsed, and I was like, that's terrible.
glass.
Yeah, and I had to watch it multiple times before I realized that nobody around them was reacting. And I'm like, that's what's weird about it. Like, it took me a while to figure it out.
I had that similar situation where it looked like I thought a lion cub was going to be eaten by crocodiles, but an eagle took it back to its dad, and I'm like, what?
We need to figure out is how they video it.
That's
Yeah,
what
yeah,
so I'm like, that must be
Does
weird.
anybody have questions for her? No? Thank you! Alright, you guys take it back to your lab. You got you've got about 35 minutes. That's a lot. Oh, yeah, by the way, if you were not here last week, hold on. If you were not here last week, you can come up here and get a lab that we're doing just for fun on the side. It's a dragon lab where you can take alleles to create your own dragon and eventually we'll breed him. You can do that whenever, like on your own time. Like we may come back to it a little more next week, but this week I want to give people a little more time for their lab. And you guys get phase 2. By the way, this team is getting phase 2. Woo! She's excited. All right, go for it. You can go. Let me give them their great job. That's really
I hope I didn't talk too
No,
fast. I have a bad habit.
They're used to fast. They're used to me, so they're.
No,
really well.
I used to be nervous presenting in class because there was so many like decorating and all that.
What number are you?
We wanna make sure that
I just one word, it's maybe like type.
it can be a good tool